All right, so let's start. It's 2 minutes past 1, and I'll try to keep my time as well. So welcome everyone. I'm very happy to see you and to have you on board on today's Outreach Week session with the topic towards precision and translational medicine. And I'll be your host. My name is Päivi Östling. I'm a cancer researcher, precision cancer medicine researcher in my everyday life, but I'm also the scientific lead for precision medicine team at SciLifeLab. So I'm pretty sure you're quite well familiar with SciLifeLab as an organization. I just want to remind you all that the main visions of SciLifeLab is to enable Sweden as a world-leading nation in life sciences. Okay, make some trouble. And our mission is really to enable research that otherwise would not be possible through the national infrastructures. There are 3 main pillars at SciLifeLab, meaning the infrastructure, of course, then the researchers that use the infrastructure, and then the newest pillar, which is the data-driven life science. And today we hear more about the link between the infrastructure and the research, meaning the technology-driven life science, and touch upon the data created by this. The infrastructure consists of 10 different platforms that you see listed on this. And these are actually quite big platforms consisting of 36 or even more units. And most importantly, we have hundreds and hundreds of technology experts working at each platform and unit, performing their best, providing you with the best possible service available. That's why we also need to highlight the SciLifeLab community, which is really that you can find the best expert on the specific method that you want to use anytime and in any platform that you contact. So there's really a big heart within SciLifeLab, I feel. Besides platforms and units, we also are organizing our activities and capabilities, and I have listed a few of these, such as pandemic laboratory preparedness, precision medicine, and planetary biology. And these are really across platform capabilities. Then there are also bigger platforms that have already within platform help for many methods within one platform. But if you look closer across the platforms and specifically on precision medicine, this is really about building the bridges between the different units and help the user to engage and conduct precision medicine studies in SciLifeLab. This precision medicine capability is pretty new. We started with a small team in June of last year, and our task is to really identify the main strategies and action items, meaning the bottlenecks of how can we improve on doing precision medicine studies at SciLifeLab. And together with me in this team, I have Janne Lehtiö and Åsa Johansson. So our task is really to bind the 3 pillars together for precision medicine and to link to the data-driven life science program and genomic medicine Sweden. So these technology-driven life science capabilities, of which one is precision medicine, links the infrastructure and the research. But of course, nothing happens, or all of this activity will create a lot of data, and that links us to data-driven life science. As you all know, technology is really the backbone of the paradigm shift that precision medicine is creating for medical sciences. And you know, this is an evolving journey. It's never ending. It's not enough when we have implemented genomics, there will be the next method and so on. So our team's task is then to enable the platforms to do this journey towards precision medicine. As we all know, precision medicine is a very, very wide concept, and it influences the whole society. And that's why I wanted to use this figure from the UCSF to really demonstrate that there are multiple stakeholders and the society as a whole for precision medicine. However, our capability is really about building the knowledge networks and using our technologies to increase the knowledge-enabled discovery. A similar journey from discovery research to routine has already been done through our clinical genomics platform in creating Genomics Medicine Sweden. And now the question on our minds is that how can we make this journey for the next method and really bridge this moving from proof of principle to translational and clinical research and trials. We believe this will need to happen stepwise, and here our capability is mainly focusing on identifying technologies that are mature enough to take this leap and to hopefully initiate in many proof of concept projects across platforms. That will enable us to understand what type of sample flows are needed and what type of data this will create. Of course, as within any human involvement in research, there will be a need for a defined ethical framework. And to really truly implement precision medicine, we need to have available treatments at the clinic. This will require policy and funding changes in Sweden. And these are, of course, very big items that are not directly, but indirectly linked to our work. So far, we need to ask our platforms as to what is the technology readiness level of your method. So which methods are mature enough? What type of samples are needed and how much? What's the turnover time of your assays? Usually within a clinical setting, you need to make decisions pretty quickly. And can we really rely on the results? Are they reproducible and of high quality? And is the analysis also automated? And so on. So these are some practical questions that we will need to work with the help of our platforms to define. However, you have to remember that these methods that are all listed in this visionary slide on precision medicine, where you would actually have longitudinal samples and different types of samples, are available at SciLifeLab. So this is not science fiction, but we do have the methods and we have the experts that know these available at SciLifeLab. However, it is not a simple task as to use one method, but to really link this to create an ecosystem to maximize data content on these valuable human samples and to create the maximal benefit of them. If we move forward, today we have these speakers, these 5 speakers that have already used the platforms available at SciLifeLab towards precision medicine in different types of diseases and in different stages. And I very much look forward to hearing their examples on how they have found working towards this translational medicine using our platforms. In our session today, we have 7 different platforms highlighted as listed here. You can see these boxes. And we're moving from genome to transcriptome, metabolome, microbiome, epigenome to cellular profiling and tissue profiling, including also functional testing with drugs. And of course, to make sense of it all, bioinformatics and integrative multi-omics analysis is really key. And we'll hear some examples of this today. So these are really examples of how we aim to move towards precision and translational medicine. We're not there yet, but we're preparing our organization to be able to be more helpful in this. So we're really going all the way from biochemistries to single cell to spatial and functional profiling of human liquids and tissues. And here I have one slide of each of the 7 platforms, and I don't intend to go through them in any detail. I mainly want you to notice the contact details for each platform, which can of course also be found on our web pages. And I will make these slides available in case you need. So we have a very big genomics platform, and specifically a clinical genomics platform, which really works on real world clinical research with returning data to the clinic on an everyday basis. We also have a metabolomics platform that will be presented today, and here you have their contact informations, as well as clinical proteomics and immunology. We also have a spatial and single cell biology platform being highlighted in one of the talks today, as well as, let's see, next slide, please, here, the chemical biology and genome engineering that provide drug repurposing libraries for functional testing. And these are some of the examples available. And you have the contact details here. And not to forget NBIS, of course, our national bioinformatics infrastructure that do help us in many different types of analysis included here. And you can see the information details here. And with that, I would actually like to conclude my introductory session and introduce Eva Hellström Lindberg, who is our first speaker today. She has a topic of towards precision medicine in myelodysplastic syndrome. And she's a professor in hematology in Huddinge at Karolinska Institutet. And she has a long and great career, and Eva, we look very much forward to hearing you present today. You will have 15 minutes, and I will inform you when there's 5 minutes left and then 1 minute left. Okay, so, and for the audience, sorry still, please do write questions in the chat. And if there's time, I will ask them from Eva. If there's not, we save them for the later QA session. Okay. I will stop sharing, and Eva, go ahead, please share.
Okay, thank you very much, Päivi. Thank you for that nice introduction. I'm happy to be here, and I'm happy to take you through some of the things that that we have done during the last years. My title of my talk is Towards Precision Medicine in a Disease Called Myelodysplastic Syndromes. And for those of you who are unfamiliar, I have one slide to explain what that is. So precision medicine in blood cancer is actually always weighing disease characteristics against patient characteristics. And what do I mean with that? Well, in the disease characteristics, we have, of course, inherited conditions. Sometimes we have obviously germline inherited disease, but can also have predisposing genetic conditions to later, for example, myeloid malignancies. And then we have all the tumor characteristics. I mean, the genomics, the complete genomics, as we have heard, but also biological aspects. On the patient side, we have age, of course, organ function, how old and tough is this individual, not mentally, even if that matters too, but how well will this body tolerate different types of treatment? What are the patient's preferences? That's very, very important. And if we have a treatment, what is the expected effects of the treatment, and what is the expected side effects of the treatment? And of course, this always cooks down to the life situation of that individual. And for 30 years, we had the most fantastic example, and that is the imatinib for BCR-ABL positive. Sorry, this is my post-COVID cough, you have to bear with me. Positive chronic myeloid leukemia. And that was established like in 1990. And today, instead of a median survival of 3 years, we cure about 90% or 90% of patients with CML. It will, they actually have normal life survival, but expectancy. But that is not what we see in the majority of patients. Most other blood cancers are much more complicated and much more difficult to target, such as MDS. This is a heterogeneous group of blood cancer originating in the hematopoietic stem cell, and due to that specific effect, it's almost impossible to cure by chemotherapy or any other type of treatment. So most patients with, I have my coffee. So you can only cure MDS by actually providing the patient with a stem cell transplantation. The disease is characterized by ineffective hematopoiesis, by progressive cytopenia, and it has been defined by morphological changes in the bone marrow and or the presence of immature precursors in the bone marrow. But the prognosis is highly variable between subtypes, between almost normal to an expectancy of perhaps less than 1 year. And I usually quote this patient that I met some 15 years ago, some years before the publication of the first whole genome sequencing of a patient with acute myeloid leukemia. And this patient, who was a bit provocative, he told me, Eva, I want you to take my bone marrow and investigate every single gene in it, and then decide what treatment I should have. I think this is a fantastic statement of a patient that was not really a well-educated person, but he understood the context of precision medicine. And now, 6, 7, perhaps 8 years ago, we formed the IWG, International Collaboration Project for MDS. And we have together sampled over 3,000 samples, curated correct samples. I think we sampled almost 6,000 at the beginning, from 13 countries and 25 centers. And 1/3 of these patients is actually from Sweden, the majority from Karolinska Institute, and about 1/3 of these from a biobank that we organized in Sweden. And looking at this patient, this is an ordinary, list of what we find. We found 108 genes recurrently mutated, over 5,000 unique driver mutations, and a total of 11,000 mutations all over. The most common feature in MDS, and that is the problem with MDS, or has been the problem, is that the most common molecular event is a point mutation, and the majority of these are not hotspots. So there are hotspot mutations, but the vast majority are found in recurrently mutated genes, but they are individual variants, most of them. This is very different against acute myeloid leukemia that we will hear the next talk about, where you have much more recurrent events, balanced translocations, and also hotspot mutations, even if the part of AML looks as MDS. So this IWG network then has come out with one major publication a couple of years ago, or 1.5 years ago almost, and that is the notion that it depends in what way you are TP53 mutated. It was considered that if you have a TP53 mutation in MDS and in acute myeloid leukemia, it's actually bad in any way. But what we showed in MDS was that it's indeed so that the double hit TP53, that means either 2 mutations or a concomitant loss of chromosome 17 or 17p, or in that case, loss of heterozygosity, actually implies a much worse outcome. And this is now the group that is in the focus for therapeutic attempts. This is probably the most difficult disease we have to treat. Another delivery is now submitted and will hopefully soon be published, and that is the IPSS-MOL, the new international prognostic scoring system for MDS. And all blood diseases have these IPSS systems. Most are called IPSS-R, revised, but we have actually integrated all these genomic data in the previous IPSS-R, and we now have very soon an international algorithm and a calculator that we can use to classify and help our patients, and also with time choose the optimal treatment. And this is what we use in the clinics in several years. And before that, we had since 2016 another smaller panel. This is the clinical tool we use for all patients with MDS and with AML. So I think we are very far ahead on understanding our patients. So in my research group then at Flemingsberg, We also work with integrative genomics, and here we have had fantastic help of SciLife. So we have the 1,000 patients sequenced at Memorial Sloan Kettering Cancer Center by our collaborators there. They are fully curated and annotated by us, and we have our whole own sort of cohort back in our hands. In about 500 of these, we managed to select CD34 positive selected bone marrow cells, and we have made a transcriptome analysis at SciLife in Stockholm, and using then the SmartSeq2 technique that allows us to link it back to other studies that when we use single cell sequencing. I will not talk about these studies today. And we can then add on methylation, ATAC-seq, proteomics, which we also are working with together with SciLife. And then we can combine that with the perfect outcome data we have in Sweden, with the treatment knowledge. We have actually every single transfusion given to every single patient in the database. And we are starting now a project to really understand germline factors in a better way than has been done. And then we come to personalized medicine, bringing in also the preferences of patients' personalities, as I said before. But this really gives us a power. So I will actually conclude my talk, and I will by talking a bit about one of the prospective clinical trials that we have that use precision medicine in a different way. That is to really find minimal residual disease. And as I said, the goal, patient with MDS that is young enough and fit enough, we aim them to undergo stem cell transplantation because that is the only curative regimen. However, even if we do so in clinical practice, about 30% relapse, unfortunately, and around 15-20% over the years experience non-relapse mortality. So it's not an easy treatment to take. So we decided in a pan-Nordic study to actually start to do something about for the cohort of MDS patients. So part one of this 14B study was a non-interventional study. We collected in a total of 286 patients between 2016 and 2020. It was hypothesis generating, it was unbiased, and we just wanted to develop and evaluate a personalized MRD method based on NGS as the original investigation. We are now soon to start the part two, that will include 200 patients, again at the Nordic level, that will prospectively enroll patients with a real life measurement of MRD. And we will use the novel MRD method for routine intervention with the aim to reduce relapse risk. And everything that has to do with allogeneic stem cell transplantation is to learn how to deal with the clone and the risk of relapse, and the immune system with its possibility to cure the disease, but also with the possibility and risk for actually killing the patient. And this balance is what we would like to improve with this study. So for the first part of the study, we actually screened 286 patients, and included for MRD analysis was over 200, which was actually way more than we thought. This part of the MRD was developed at SciLifeLab in Uppsala. Some patients were excluded. I mean, some died very early, some didn't have anything to make a probe and primers for, and so forth. And some were actually lost for follow-up. But we have more than 200 MRD patients. Among these, with 32 patients with non-relapse mortality, with 52 experiencing relapse, and with 137 in remission. And there's a lot of data, but I will just tell you some of these. So this is then the relapse-free survival, ending up around 60% over 5 years, which is a fairly good plateau according to international standard. However, if the patients get a clinical relapse, the overall survival decreases very much down to a plateau of 20%. There are still some patients that we can rescue, which is good, and that is also proof of principle for the coming study. But following a relapse, outcome is usually very fatal. And this is then when we compare, the orange curve here, are patients never experiencing a positive MRD. That is around 70% of all patients. Some of them, as you see, they will succumb from non-relapse mortality. And the possibility of the next study is also to be able to actually identify the early MRD negative patients and perhaps be a bit less tough with the immune component in their treatment. The blue curve are patients that at any time point experiences an MRD. And as you can see, the survival in these two cohorts is vastly different. And this motivates us now to follow up with the Nordic NBIS study part 2. And what Lucia Cavalier and her team has done in Uppsala is to take this and develop into a real-time analysis. And I think the challenge here was to realize that this could actually be done in spite of almost complete lack of hotspot mutations. So we send an NGS before transplant, some weeks before. They have about 2 months to develop probes and primers for each of the mutations that we find, and then test the assay against the normal control material. This sounds complicated, but it's actually not that difficult. And the good thing with the digital PCR is that once you have the assay, it's quick and it's actually very cost effective. So this is now what will happen. We will enroll patients from like April or so, and we will actually put them through this test. And in case of a positive MRD, the clinical team has a number of routine actions to choose from, like tapering immune suppression, donor lymphocyte infusion, or the drug azacitidine. And then we will compare this part with the previous part and see whether we actually can save some patients from relapse. So with that, I think I have I'm within time, and this is the NBIS group. We have the collaborators, other independent research groups, including Seisho Gava from Kyoto, and also who is also a guest professor at KI, and also all the international collaborators in the Nordic NBIS group. So thank you very much. I will then stop share my screen so I can take questions.
So thank you, Eva. Very interesting presentation, and this is really, truly, in clinic research being done. And I actually had a question, but you kind of answered it. That your turnover time then for your digital PCR assay is 1 week to be able to make a clinically relevant decision, right? That's quite impressive.
Yeah, so we think that if you take the sample on Monday or at latest Tuesday morning, you will have the response before the weekend, and you can take a decision and start to intervene on Monday the following week.
So what volume of sample? I mean, I know PCR doesn't use a lot of sample, but I was imagining that maybe some other analysis possibly could be included besides digital PCR in this type of a flow of material. Would you foresee any other type of technology that might be of benefit for these patients already here?
I mean, definitely. So DNA will, of course, be stored in this. So we can do everything. I think one thing that we also do is that we aim to store vital frozen cells, because what we need to understand is if the DLI effect, if the immune effect is actually it's better against some mutations and worse against some mutations. So we have all the possibility to proceed with detailed molecular biological and cell biological studies. So what I showed you now is what we actually will do in real time. But in addition, we will have a huge biobank that allows us to address various questions that may come later on.
Yeah, thank you. That's kind of the point I wanted also the audience to get, that many of these studies start with retrospective cohorts and studies when you optimize things to move on to exactly like you have now, prospective real-time action. We have a question in the, or actually two questions in the chat from Isato Sar. So I'll just ask the first one and see if we have time for both of them. With regards to MDS, how do you overcome diagnostic difficulties due to concurrent treatment or disease?
Sorry, which question was that?
It's
how do you overcome
diagnostic
difficulties?
Yeah.
Well, I think with the new molecular, I mean, MDS has been a difficult disease to diagnose because it's blurred, it's cytopenia, the bone marrow may look strange, but with the molecular diagnostics that we nowadays are doing, I think we are much better actually at differing between aplastic anemia, acute leukemia, and MDS. So I think the diagnostic problem is not as big as it was some years ago, if that was the response.
I assume so. And there is a follow-up question then from the same person. Is there a gold standard / clinical algorithm for the diagnosis and treatment of cytopenia with clonal hematopoiesis not fulfilling MDS criteria? Do you have
that's a very good question. I could speak about that for a long time.
Sure,
I think what is now called CCUS, clonal cytopenia, will with time in the new WHO classification be sort of a subgroup in the MDS chapter. And we do not know how to, I mean, the concept is that these patients do not have cytopenia, or at least not clinically significant cytopenia. The question is whether you should do anything to try to halt the clones from expansion. And I think that depends again on the type of mutations you have. We have a longitudinal CHIP program together with Sten Erik Jakobsen, who is the lead of that. And we actually resampled patients with CHIP and with CCUS. And over the years, very little happens actually. And what we have also another project, we have traced back clones in MDS 30, 40 years. That has also been done for myeloproliferative neoplasms. So the question is very good, and I think this is one of the key focus for research around the world. But you have to be very careful when you treat CCUS or CHIP, that the cure is not worse than the clone, but these are also prospective questions for research. And then I got a question, and I would say that I wouldn't like to share my slides because, I mean, like more than half is actually data, unpublished data. So with respect to my collaborators,
That's fully understandable, Eva. I'm able to share my introductory slides because they don't contain data and mainly contact information as such. But with that, I would like to still thank you, Eva, for a very interesting talk and introduce. So now it's 12 o'clock and we continue with our towards enabling precision and translational medicine session of the SciLifeLab Outreach Week. Our next speaker is Kristoffer Sundling. He's a researcher at the Department of Medicine. Hi, Kristoffer. At Karolinska Institutet as well. And as before, I keep your time. I'll alert you in the chat when you have 5 minutes left and then 1 more minute. And if there's time, we'll do questions. So please go ahead, Kristoffer.
Thank you. And thanks a lot for allowing me to present in this forum. So I'll change gears a little bit. I'll talk about infectious diseases and actually about more or less the plunge that we've done into malaria immunology, which is quite complex. And I must say that SciLife has been really helpful in allowing us to do this plunge. Let's see. Right. So malaria, it's, it might be something you heard a long time ago, but, you don't hear much about it anymore. But it's still a quite large problem. So just some basic information. It's a parasite that normally resides in mosquitoes. It's transmitted to humans via saliva, where it goes into the liver, and there it sort of develops and changes its antigenic structure more or less into a blood stage parasite. So the parasite sort of has several different stages, and at any given stage, it more or less have 1,000 different proteins expressed. So as you can imagine, it's quite difficult for the immune system to target necessarily the correct proteins or to find the perfect ones to use for vaccines and so on. So vaccine design is still a very relevant problem. Like I mentioned, it's still a big problem in the world. We have several different species. The falciparum is the most prevalent. It's about over 200 million cases globally. Most of them happen in sub-Saharan Africa by this falciparum variant, and over 400,000 deaths, most children under 5. So very severe disease for small kids. And this is more or less what you could expect to see when it comes to disease severity in an endemic setting, where it's mainly the young kids that get the severe malaria. And then as time progresses and they get malaria several times, you get milder symptoms, and in the end, maybe even asymptomatic. So the interesting part, I think, here is partly that you don't actually stop being infected. You just get less severe symptoms from it. So it's a very adapted parasite, in a sense, to cohabitat with us. But of course, we don't want to have this severe malaria, but we really want to understand it. So it's mainly the blood stage that's responsible for this clinical presentation. And when you reach where you're still infected but don't have symptoms, you call it clinical immunity. And if we can't reach sterilizing immunity to stop the parasite completely, at least we want to reach clinical immunity early on. So also over time, as you get progressively more infected, you see a broadening of the adaptive responses, so more antibodies, and also a more diverse antibody response to different antigens. But at the same time, you actually see a sort of decrease in the function of the innate responses. To the left there, you see gamma delta T cells, which are responding quite vigorously to malaria. But for example, if you had malaria several times, you can sort of see how they respond less and less. And recent study also show that it's quite similar for monocytes, actually. So over time and in adults, they're actually not responding very much anymore. And the PCA plot there to the right just shows stimulated monocytes from kids versus adults. So very different transcriptomically as well. So we're very curious to sort of understand both this innate part and the adaptive part, and actually how they belong together. But I come from the B cell field, so I'm very interested in B cells to start with, at least. And in a malaria B cell response, you kind of have this large amount of pro-inflammatory cytokines. It's a lot of pro-inflammatory cytokines in malaria, probably causing a lot of the symptoms. But these pro-inflammatory cytokines, they also drive the B cells perhaps away from what you would consider a normal response to more short-lived plasma cells. And also these fairly recently described atypical B cells, which we don't know very much about yet. So we don't really know if they're good. What purpose do they have? Where do they really come from? So I think that part has maybe been a bit more described now, coming from memory, and I'll show you a little bit more about that. But still very unclear what they actually do there. So this was how more or less our project started and how we got into contact with SciLife, because we wanted to sort of take a broader view of the immune response during malaria. So we contacted Peter Brodin, that was probably in 2016, and performed mass cytometry on some individuals at several time points after acute infection to see how the immune response developed over time. And especially looking at B cells, I saw these CD11C positive cells. And CD11C is a marker you would normally associate with dendritic cells. So I was quite curious about that. Then using flow cytometry to look at sort of a larger cohort, we could see that individuals that have been exposed to malaria several times versus those that sort of got malaria for the first time, they both could form these CD11C positive cells, but it was much more of them in people that had malaria before, and both of them expanded compared to controls. Looking further at these cells, we could see that Ki67 positive cells, which is basically cells proliferating, so they're responding to the infection, almost 80% of those expressed CD11C. So it really seemed like CD11C was this marker associated with cells responding to the infection itself. And we could see here that a lot of those cells in the primary infected individuals were actually IgD positive, meaning that they haven't class switched. So they're more likely coming from a naive B cell compartment. So now we could sort of learn a bit more here how memory cells responded and how naive cells responded. But we also found a large heterogeneity within these CD11C positive B cells. So we really wanted to look more into what they both were composed of, different types of cells, and also potentially if we could see transcriptomically how they evolved over time. So I contacted this eukaryotic single cell facility at SciLife, that was in 2018, I think, about doing 10x sequencing on these cells, and also look at the B cell receptor sequences to look at clonality and affinity maturation and so on. So they did the library prep for us and sequencing, and we could see again here that we saw cells with a memory phenotype, some kind of intermediate stage, and then an atypical phenotype, or I guess transcriptional type. And this is something that we're still actually looking into. I have a fairly new PhD student, Alan, working on this. And looking at the VDJ sequences, so the B cell receptor, we could see that many of them indeed were sort of unswitched. They were not mutated very much. And the ones that had switched had mostly IgG3, which is one of these inflammatory isotypes. So, but looking at those B cells wasn't everything. Within the mass cytometry panel, we also had several markers for other immune cells. And when looking at these primary infected versus previously exposed individuals, we saw that the sort of populations or clusters didn't overlap in many places, both for monocytes, B cells, as I talked about, NK cells, and T cells. So we kind of saw differences in a lot of different compartments. And these differences kind of also translated to clinical effects, such as previous exposure, they had less parasitemia, they were also in the hospital for a shorter time. So there was an impact on this, and we wanted to sort of connect what's the, how does the immune system sort of connect to the clinical presentation. So here we also contacted SciLife because we wanted to look at the inflammatory proteins. So we contacted the plasma profiling facility and wanted to use O-link affinity proteomics using this inflammation 92 panel, which would give us a good overview of whatever soluble inflammatory responses there were. And so this went completely fine. And we also had this FACS data from before. So now we wanted to sort of do an integrated analysis of those. And that was done by my PhD student, Julius. And he used multi-omics factor analysis, this Mephisto, which also can use time as a covariate because we had looked at these samples over a full year. So he did this analysis and saw how sort of time was an important factor in how the immune system looked. And just looking at the different sort of immune cells and cytokines and so on that were changing over time, we could draw this kind of landscape or map of what was happening. And also then we could start comparing what's different between the primary infected versus previously exposed individuals. So there are a lot of differences. And some of the really key ones, which I brought up early on as well, were associated with these adaptive responses versus innate responses. So for example, a lot of these early cytokines correlated quite strongly with, for example, gamma delta T cell responses. Also monocytes, we saw some changes in. And I think quite interestingly also with antibodies, when we repurposed an antibody data set. So based sort of on both the adaptive responses, the innate cells, and sort of the antibodies, we could, I think, draw kind of a map of what we think is happening in the primary infected versus the previously exposed individuals, and how sort of tolerance starts to develop, how symptoms starts to reduce as the innate response gets lower at the same time as the antibodies get sort of more and better. So this is currently in revision with quite positive reviews. But in the next step, we were really curious to see, so we saw all these changes, but we wanted to see, does that make sense transcriptionally as well? And so we wanted to generate this transcriptional landscape of malaria as well. And here we collaborate with Peter Brodin, and we've done sequencing at NGI at SciLife. And we use this BD antibody sequencing or Rhapsody system, where you have antibodies with oligo tags. So you can basically get a protein profiling at the same time as you get the transcriptomics. So we included this 29 marker, quite heavily focused on B cells, but also on other immune cell subsets. And these panels containing 399 inflammation-associated genes. And the advantage of doing that compared to full sort of single-cell sequencing, transcriptomic sequencing, is that we need much fewer reads per cell. So instead, we can actually run many more cells in the same experiment. So this is an ongoing sort of study, and initially here we've run 4 individuals at 3 time points, so 12 samples, and sequenced about 100,000 cells. And this picture just shows sort of how the immune landscape looks in these individuals. And now we'll start really looking into how it changes over time, how that's associated with primary infection or previously exposure to parasites. And if we can repeat what we've seen in monocytes, gamma delta T cells, and the B cell compartment. So this is a sort of project that's run both by Julius and Alan for different parts. So to summarize, I think we've used SciLife in a lot of different capacities that have really been key to sort of drive this type of research forward in our understanding, I think, of how malaria immunity works. And which is a really important step when we actually want to aim for vaccine design in the end, and understanding how potential when we treat and how we treat and so on. So it's been a large collaborative project. A lot of people have contributed, as shown here. And especially I would like to thank the study participants. So thank you for listening.
Thank you, Kristoffer. Nicely kept time. You even had 1 minute left to go. Very interesting and nice to see another example than cancer on how to really utilize these precision medicine adaptable tools. If you were to vision forward as to when would you make an impact, or would you need to make an impact on malaria treatment? Which method or how would you go about doing that?
So I think one part is basically understanding how this clinical immunity to malaria develops. Because currently, some studies have looked just at the antibodies or only at T cells and so on. But I really think this integrated approach is important, because the immune system, it's all about the system, right? So I think you really need a systems approach to actually understand it. And in my view, if you really understand how this immunity develops, then you can start to play around with it. Either by like normal conventional vaccine design, you try to identify different antigen targets, but perhaps also to play around with these innate cells. So maybe you should actually give younger kids sort of immunosuppressive treatment for this innate response. But maybe that's really dangerous to do unless you have these first adaptive responses. So I think we really need to know how important are the adaptive responses in sort of suppressing the innate responses before we start playing around with that. But
No, thank you. I fully agree. I mean, every disease are complex, and you need these integrative approaches to even be able to understand what are the next steps that needs to be taken. So thank you for a very nice presentation, Kristoffer. I learned a lot. Very interesting. And next up, I would like to introduce Shahrzad Shirazi Fard, also from Karolinska Institute. Hey, most welcome. She's an assistant professor from the Department of Women's and Children's Health. So please go ahead and share. I'll time you and let you know when you have 5 minutes and 1 minute left. And questions, please write them in the chat and we can take them afterwards if there's time.
Okay, thank you. Thank you very much for that really nice presentation. I'm going to present some of the work that we have been doing on mapping the single-cell immune landscape of human neuroblastoma. Just some background. Neuroblastoma is one of the most common extracranial solid tumors in children, and it arises from the neuronal crest progenitor cells in the sympathetic nervous system. It most commonly presents with tumor masses in and around the adrenal glands. Every year, approximately 20 children, most of whom are below the age of 2, are diagnosed with neuroblastoma in Sweden. When it comes to childhood cancers, the overall survival has increased over the years and has now reached a 5-year survival rate of 85%. However, when we look at neuroblastoma, the red arrow here, the 5-year survival rate has stagnated, peaking at 75%. The prognosis of neuroblastoma depends heavily on the disease stage, where the variation in the survival rate among patients reflect the clinical heterogeneity. Neuroblastoma has a wide range of clinical outcomes, from spontaneously regressing tumors to highly aggressive and metastatic cases. Whereas patients with low-risk disease have a favorable prognosis with more than a 90% survival rate, the 5-year survival rate of patients with high-risk disease is still below 50%. So how can we improve this? Recently, multiple studies in neuroblastoma have pointed to a critical role for the immune system in both prognosis and response to treatment. Although childhood tumors are traditionally considered to have a low infiltrated immune response and are therefore considered to be immunologically cold, there are studies that have demonstrated the presence of tumor infiltrating lymphocytes, including T cells and NK cells in neuroblastoma. However, B cells have been reported to be rare and mostly undetectable. Moreover, tumor-associated myeloid cell infiltration has been suggested to predict both poor and improved clinical outcome. And therefore, our knowledge of the different immune cells in neuroblastoma is limited and have either conveyed conflicting results or focused on the single cell type. Therefore, understanding the tumor cell composition will be crucial for the development of novel therapies and to further improve survival of children with neuroblastoma. The goal of this project was therefore to unravel the immune cell landscape of neuroblastoma. When trying to unravel the tumor composition, the standard approach for years have been bulk RNA sequencing, where tissue is kind of grinded up and you get an overall feel of what's in it, just like this milkshake. This is a very powerful method for practical reasons. Even though you might not be able to get information on the main ingredients or cell types, there is loss in sensitivity, and cells that make up a smaller population will be lost. To circumvent this, the single-cell approach has been developed, and compared to the milkshake, we are here able to get more detailed description of what's actually in the bowl. Using the single-cell approach on tumor tissue, we were able to catch more information and identify even small population of cells. We believe that this was really of importance since comprehensive immune cell overview at the single cell level is still missing in neuroblastoma. Neuroblastoma samples from 17 patients were obtained. The tissue was dissociated, generating a single cell suspension, and that consisted of approximately 46,000 cells, which was then used to comprehensively characterize the immune cell landscape. A joint analysis of all samples was used to create a complete cellular map, including tumor cells, stroma cells, and the immune cells. First, we were able to identify 3 subpopulations of malignant tumor cells. These were the mesenchymal, Schwann cell precursor-like cells, and the adrenergic tumor cells. Furthermore, our initial analysis of the immune cells revealed a rich repertoire of the main immune subtypes, including myeloid, B cells, T cells, and NK cells. And these consisted of 10 clusters in total. This could be further subdivided into 27 subpopulations, including distinct subtypes of myeloid cells, such as monocytes, macrophages, and myeloid dendritic cells. The single-cell RNA sequencing data is quantitative, but it did not let us know where anything is in relation to each other anymore, so we kind of lost the orientation to one another. On the basis of the results from the gene expression profile, we next assessed the spatial composition of the tumor samples using the multiplex immunostaining called Codex. The Codex technology is provided by the Spatial Proteomics profiling facility at SciLifeLab, and the technique allows for a highly multiplex analysis for upwards of 40 proteins, and they use cycling detection of DNA barcoded antibodies. This was done in order to characterize the spatial resolution of the immune cell landscape. So here we have an example of one of the tumor tissues that we used. This sample was labeled with a 9-marker panel, including CD3, CD4, CD8, CD11C, CD15, CD19, CD45, CD278, and 279. And these included markers for T cells, myeloid cells, and B cells. And besides markers for immune cells, DAPI was used as a nuclear marker, and thereby labeling all cell types. And DAPI is always displayed in blue, as you can see here. And here you can see a magnification of 3 regions in the tissue, showing immune cells positive for markers for myeloid cells in region 1 and 2, for T cells in region 2, and for B cells in region 3. These results were quite intriguing, showing how close the different immune cells were to one another. The different immune cells could also be further characterized when combining even more antibody markers. For example, previous studies have indicated that tumor infiltration by T cells is especially low in high-risk neuroblastoma, with CD3 positive cells making up approximately 5% of the total cells in the tumor microenvironment. Similar results were obtained in our stainings. However, we were also able to identify small clusters of cells located within the tumor mass. And these clusters, as you can see here, expressed markers for T cells, including CD3, CD4, and CD8. And we were also able to identify subpopulations of CD4, CD45 row double positive cells, indicating memory cells. And we also saw proliferating T cells expressing the proliferating marker Ki67 and CD4 or CD8. So even though the total number of the infiltrating T cells seems to be low in neuroblastoma, there might still be a prognostic value in the localization and the organization of the T cells. Further studies are therefore needed in order to identify a possible correlation between the spatial contexture of the T cells and survival in neuroblastoma, similar to findings in other cancers. In fact, I think there was a recent paper by Hammer and colleagues showing that the spatial contexture of lymphocytes was a prognostic factor in triple-negative breast cancers. Furthermore, whereas T cells have a direct tumor cell killing capacity, their function depends on the antigen presentation and co-stimulation by dendritic cells, which are therefore central to anti-tumor immunity. Using the Codex method, we were able to detect antigen-presenting myeloid cells. These were CD11C and HLA-DR positive in the tumor microenvironment. Importantly, it has been reported that neuroblastoma can induce dendritic cell dysfunction by modulating the antigen-presenting expression of HLA-DR. However, we saw high expression of HLA-DR positive cells, indicating a functional ability to actually stimulate an effective anti-tumor response. Next, we further confirmed infiltration by B cells using the Codex method again. Here we were able to detect CD19 positive B cells and antigen-presenting HLA-DR positive cells. In recent studies, it was demonstrated that B cells and the presence of tertiary lymphoid structures in adult tumors, such as melanoma and sarcoma, correlated to improved response to immunotherapy. We were in our samples able to detect a B cell aggregate, and you can see that in red here, combined with antigen-presenting cells. This was seen in one of our samples, supporting the existence of lymphoid structures even in neuroblastoma. When we did survival analysis, we did detect a trend in the presence of lymphoid structures with survival. However, the results were non-significant, and we think that's because we didn't have adequate samples to actually study. However, when we did survival analysis on the bulk RNA sequencing data using the gene signatures derived from the single-cell RNA sequencing data, we were able to show that high expression of immune-related genes was significantly correlated with improved survival in neuroblastoma. So to summarize the results, we were able to identify 10 main different immune cell populations, and this included 27 subpopulations. And we saw infiltration of the different immune cells, and this was validated using the Codex, where we actually saw both clusters of immune cells, but also formation of lymphoid structures. And we also saw improved survival, which was correlated to high expression of immune-related genes. And this is the current status of the project. We have actually been invited by the editors of Cell Report Medicine to submit our revision, and we are planning to do it during the next couple of days. So I would like to thank all of the groups and people that have been involved in this project, and especially the Barjavno lab, which has done a lot of the single-cell RNA sequencing data. And I would also, of course, like to thank SciLifeLab for providing the Codex technology. And I would like to thank you all for listening. Thank you.
So thank you, Shahrzad, a very interesting presentation again. And I'm curious to learn whether you have any plans to now link your smoothie to your fruit bowl or the nice looking cake, meaning that do you see any benefit then in actually integrating or using both the single cell RNA and the spatial contexture that you were able to identify?
So for this first paper, we did kind of a validation more. So we kind of showed that the technique worked and that we could get the stainings to work. In the next one, we are actually currently collecting more samples to get the low risk, intermediate risk, and high risk samples. and then make profiling them with using the codex and hopefully correlate that to treatment outcome. So that's kind of the next step. But I think I mentioned that we get maybe 20 cases per year. So it takes a couple of years to get the samples that we need.
And then a follow-up as to how quickly do you think, if you were to do this in a prospective manner for these 20 patients or so, do you think that's feasible using this type of technology that you have in your study?
Well, yes, could you clarify? Sorry.
No, so actually doing it in a prospective way that like, I know there is a lot of analysis going into the spatial or the spatial contexture in a way that how quickly do you think you could actually turn this into a clinical tool is my question.
Well, if you want to do it per patient and not kind of collect and do all of them together, from the start of once you have your panel that you want to do and you have your samples, I think one round of this took 10 hours. So if you give them the samples in the morning, you will have the results the next day. So it goes really fast.
Good to hear. And in the interest of time, please write your questions. I see that you have at least one question from Kristoffer in the chat that you would can answer to in writing. And thank you once more for your very nice presentation. And next up, we have Ulrika Andersson-Hall, who is researcher from Göteborgs Universitet, University of Göteborg. So please, Ulrika, go ahead.
Thank you very much, Päivi. So I will try to share my presentation. There we go. Huh? Oh, there we go. Okay, so great. So now we'll change subject a little bit. I will talk about gestational diabetes and obesity in our PONCH study, where we look at metabolic consequences for the mother and the child. So just a little bit of background on pregnancy. It's quite a cool metabolic state, I think. There's a lot of things happening in the pregnancy physiologically that you don't see otherwise. So just one thing, for example, is that the mother gets increased insulin resistance quite naturally. And this is so that more glucose will be shunted to the fetus. And in order to compensate for this, the mother will increase her insulin production. The beta cells will both proliferate and also increase their function. So this way, a mother can keep a normal blood glucose through pregnancy. But what sometimes happens is that you get something called gestational diabetes, which is a pathological glucose tolerance that's discovered during pregnancy. In this case, we usually get further increased insulin resistance, for example, at high BMIs, or you can also have a defect insulin response if the beta cells can't compensate. Normally, we have a combination of both of these. So in that case, the mother will have an increased blood glucose during pregnancy. And what's interesting here is that as soon as she delivers the baby, within 48 hours, her blood glucose will be back to normal. So what we are interested in, though, is that there are short and long-term risks associated with gestational diabetes and with maternal obesity. And I've just put up a few short-term risks. I'm not going to go into that now, but that's risks during pregnancy and delivery. We're more interested in the longer term, where the mother will have an increased risk of cardiovascular disease or type 2 diabetes, and the child also will have an increased risk of obesity, metabolic syndrome, and type 2 diabetes. And especially this risk of type 2 diabetes is highly increased after gestational diabetes. So these women will have a 7-fold increased risk of type 2 diabetes. About 40 to 50% of the women will get it. So this is a really interesting population that we follow in our studies. It's called PONCH, our study, and it stands for Pregnancy, Obesity, Nutrition and Child Health. It was started in 2009 by Agneta Holmeng here at the Gothenburg University. And we have a few different arms. I'm going to talk about 2 different arms where we have involved the NMR, Swedish NMR Center. One is where we look at the effect of obesity and gestational diabetes during and after pregnancy in the mother, and we look at also both the consequences for the mother and the child. And also, I'll come back to this picture later. We also have a different arm where we focus specifically on gestational diabetes and the development of type 2 diabetes after pregnancy. And I'll start with the second part because that's the study that we basically finished. Well, part of it's finished anyway and published. Well, I call this a retrospective study because we picked them up after pregnancy, but then it's actually turned out into longitudinal cohort study as well. But what we look specifically at here is the risk for type 2 diabetes development after gestational diabetes. And what we did was we tried to contact all women in the Gothenburg area who had gestational diabetes between 2005 to 2009. That's 542 women. And we took them into our lab 6 years after pregnancy and did a clinical evaluation. And we got 237 of these women in. We did glucose tolerance testing, blood samples, body composition in this bod pod, anthropometry measurements, took fat biopsies, lots of questionnaires to get a good clinical picture of these women. And this is what the population looks like then if you look at the glucose tolerance. So 139 of these women are still normal glucose tolerant, whereas about 100 have some form of prediabetes or type 1 or type 2 diabetes. So it's an interesting group because these women, when we meet them, most of them have no idea that they have that they have some form of glucose intolerance or type 2 diabetes even. So it's very important, I think, to catch these women since they're such a high risk. So the idea for us is to identify the women of highest risk and identify biomarkers for early detection. And in that way, we could take them into intervention in the healthcare system. So what we did in this study is to look at, can we predict the type 2 diabetes risk after gestational diabetes? And we have a lot of clinical data here. I'm not going to go into too much, but there are obviously a lot of variables during pregnancy that will tell us if the women have a high risk of type 2 diabetes. And also when we meet them at 6 years, there were a lot of clinical measurements that associated with type 2 diabetes risk as well, obviously. And a lot of it has to do obviously with BMI, fat mass, and so on, cholesterol, inflammation. But I'm going to show you a bit more in detail is the metabolomics we did together with the Swedish NMR Center at Gothenburg University. So this is Anders Pedersen and Daniel Malmodin, who I had a few different studies within sports science prior to this. So here in 2018, we asked them to run NMR metabolomics of the serum of these women. And I'll jump straight into some of the results. Here we just show all the metabolites that are significantly different between the different glucose tolerance groups. So normal glucose tolerance, impaired glucose tolerance, or type 2 diabetes, or pre-diabetes, you can call the middle group as well. And we try to map it out a bit more to get an idea of the metabolism. So here I've plotted all the significant variables after BMI adjustment, because obviously BMI is always a big player here. And you can see all the different squares. You have the normal glucose tolerant, impaired, and the type 2 diabetes, and how they change the different metabolites, or how they differ between the groups, I should say. Some are lower in the type 2 diabetics and some are higher. So here we can see we have obviously effects in the glycolysis, TCA cycle. There's a lot of differences in the fat metabolism in these ketone bodies. And what we actually thought most interesting in the end was the branched chain amino acids, which were highly elevated in type 2 diabetic women or also in the pre-diabetic women. And especially probably this little breakdown product, 3-hydroxyisobutyrate, is a breakdown product from valine, which was the metabolite that we found was mostly associated with type 2 diabetes, and that we want to continue with. So we've published this, and as I said, we can do interventions. After gestational diabetes, there are a few examples where if you have intervention like lifestyle, metformin, breastfeeding, you can reduce the risk quite significantly. So we want to keep looking at the biomarkers, not only to identify women at risk, But I'm also quite excited to associate our lifestyle data with metabolites. And also in some of our other studies, we have interventions. So I'm hoping that we can evaluate how follow the metabolites during interventions more to see what's happening in these women. So that's the study that we published in 2018. And then we have a current project that's more, as I said, that we're looking at the effect of both obesity and gestational diabetes, and here during and after pregnancy. And here we're looking at both metabolites and lipoproteins. What we've done here is to take, we have mothers or pregnant ladies come in. We have normal weight women, we have women with obesity, we have women with gestational diabetes. They joined the study a little bit later after their diagnosis. But they come in 3 times during pregnancy, trimester 1, trimester 2, trimester 3. And then they come in after delivery, 6, 12, and 18 months. And we also follow their children up to 6 years of age, from birth, 6 years of age. So it's quite a large prospective study that's been going on for a long time. And here we've taken the 3 groups, taken fasting serum from all 6 time points. And then we've asked Anders and Daniel again at the Swedish NMR Center this time to run NMR metabolomics and lipidomics. So I just want to show you what the groups look like. This is basic background data from different groups. We have BMI, age, parity, that's how many kids they have, gestational weight gain, how much they gain during pregnancy. And this is normal, that you see obese and gestational diabetics, they gain less weight during pregnancy. But the variables we're mostly interested in are fat mass, glucose, and insulin resistance. As you can see, obviously there's a big difference in fat mass, with the obese having higher fat mass and gestational diabetics somewhere in between. You can't really see it on this scale, but normal weight women, they gain about 5 kilos of fat during pregnancy, and then they lose it quite quickly. So that's why this particular window is really exciting, in combination with, as I say, the insulin resistance, as I talked before, which actually is the same in obese and gestational diabetics, but highly elevated, of course. And then we have the glucose, as expected. It's higher in the gestational diabetics, and it just keeps going up and up and up, because a lot of these women are going to develop type 2 diabetes. So we have a lot of, they're quite phenotyped a lot at these women. So then we want to look at the metabolic profile of these. And the aim for that part of the study is to look at the metabolic changes that differ between the groups, and specifically to associate it with fat mass and insulin resistance. And we want to look at metabolic changes after pregnancy, especially then when it comes to the declining glucose metabolism in the gestational diabetics. And then something that would be really nice as well is if we could find a maternal metabolic profile that can predict the infant birth weight or infant fat mass. So Anders at NMR Center does something like called in vitro diagnostic for research, IVDR, where he has an automated metabolite quantification, and he runs a QC of each measured sample. And as I said, this time we also used the lipoprotein subprofiling, which is quite exciting. Normally we would send in our samples to clinical chemistry and get HDL and LDL and get the values for that. That's fine. But here we get so much more data through NMR. So we get, this is just an example. This isn't my data specifically, but you get concentrations of all the triglycerides, the cholesterols, the ApoA, ApoB, lipoproteins, all the subfractions of VLDL, LDL, HDL. And then on top of that, you get distribution of triglycerides, cholesterol, free cholesterol, and phospholipids, and so on, within all the different subfractions. So this is data we're going to use, because we know pregnancy, you get a lot of changes in different lipoprotein. So that's going to be really exciting to look at. So I have just one slide of our preliminary results, which Daniel has prepared for me. So this is no PLS of the time points where it's a small text here, but the green is trimester 1, blue is trimester 2, and red is trimester 3. These are normal weight and obese mixed together for this purpose. But what we can see quite clearly is there's a lot of changes during pregnancy. And then all of these clustered here are 6, 12, and 18 months after pregnancy. So you see nothing much happens once they've gone back to the original state. And if we look at the lower graph, you can see actually what contributes to these clusters. You can see, so there's a lot of the lipids and the lipoproteins on the left, means that they're increasing during trimester 3 here, end of pregnancy. And there are a lot of subfraction dependencies. We haven't listed all of them here, but we'll look more into that. And then on the right, we have what's higher in the post-pregnancy samples, basically. A lot of the metabolites, so they actually decrease during pregnancy and then increase again. So what we do see is very large metabolic changes during pregnancy, but we still haven't really been able to pick out more subtle obesity or gestational diabetic related differences. So what we'll go on to do really is to try to associate these metabolites with maternal insulin resistance and fat mass changes. And also, as I said, with infant birth weight and fat mass. So see if we can predict, because as I said as well before, the children from these mothers, they have a high risk of obesity later in life. So I think it's really important if we could pick them up as early as during pregnancy, even to see what contributes to the fat mass increase in these infants. So that's me. And I just want to thank everyone. And yes, lots of names here, but it's the PONCH group working at the moment. We have the previous PONCHers, to call them, and then all of our collaborators, and especially then Anders and Daniel, who've been helping me from the NMR Center. Protein. Thank you.
Okay, thank you, Ulrika. Very enlightening and interesting, and nice to hear on how new types of protein, lipoprotein and metabolites can bring insights into pregnancy and risk factors of that. You do have questions in the chat on sample requirements and on microbiome. Please answer those. But in the meantime, I would actually like you to bring forward. I mean, you mentioned that you had blood samples, like serum samples, and fat biopsies, right?
Yeah.
If you were to dream as to which technologies you could actually use that would be useful for these ladies, what methods would you include in your next study? Unlimited funds.
Unlimited funds. Well, we're actually already doing a transcriptional profiling of the fat biopsies, the NGS. So that will tell a lot of interesting things. And obviously, if we could do proteomics and metabolomics on all our different samples. I also have a different study that I'm talking to Anders, where I have samples from delivery. So I have placenta, fat biopsies, and I have cerebrospinal fluid and serum fluid, all of it from C-sections. And I think if I could do a full metabolomics profile from that, that would be
very
awesome.
Interesting, very interesting cohorts. Yeah. And this really highlights the importance of well-annotated cohorts and the importance of these types of good collections and what kind of these molecular methods can really open up as of research.
Exactly. Our cohorts are quite small. I mean, if you compare to the big American studies, obviously, but on the other hand, they're very well profiled. I mean, we measure, we have a lot of time points and we do a lot of specific measurements. So I think it's a valuable cohort.
Yeah, so with that, I would like to thank all the speakers actually and open up for a short panel discussion. If you are here, all the speakers still, we could take some online questions as well. I see Anders Pedersen already raising his hand. So please go ahead and unmute yourself and ask your question.
Sorry, that was my bad. I didn't intend to.
Don't worry. I hope you found the session still interesting and worth your while, and that you have learned stuff in this discussion. So to all the speakers, maybe I would want to ask you as to when and how did you actually decide to contact the SciLifeLab platforms? So if we start with Eva.
Well, I mean, as you know, understand now, I mean, precision medicine is sort of what goes without saying for hematology and hematology. So we used, I mean, I started to use hallowed genomics like 10 years ago in Uppsala. We went over to Haloplex. We have, I mean, so we have been in the context of SciLifeLab from the very beginning. We have done some analysis abroad, and we have now continued with proteomics and with transcriptomics and with single cell analysis. So for me, it's sort of, it has been.
Yeah, that's the impression I got from all of your presentations, is that you actually have quite a long-term collaboration or integration within the projects that you have conducted in this. So maybe Kristoffer, you can go next. When did you decide, and at what time point did you had actually years in your presentation, which was very nice.
Yeah. So I think basically around 2016 it started out, and I think it worked really well. And I think in the field overall, the approach sort of to go fishing a little bit had sort of become more and more prevalent, to sort of find if there were new things we didn't know of before. So I think based on that, we got these larger data sets, and then of course you just get so much to analyze, right? And then you want to start connect them together. So from that, I think we've just expanded our use of these facilities. And now we even have one of these collaborations with the Human Genome Atlas that's going to be like a human disease atlas, which will use one of these Olink Explore 1500 panels or something. So it's all just expanding.
So then maybe I could twist the question around a bit as to, so you have all gained help from platforms at SciLifeLab, but what is then really the computational burden, and how have you resolved it? So do you have bioinformaticians in your own groups, or have you used a national platform for help? And maybe we take Shahrzad to answer this first.
Well, for this first round of stainings that we did with the codex, actually I sat down and learned how to do everything because I thought it was kind of important to know. So I did not reach out to any further SciLab facility. Instead, I kind of learned it myself. But for the future, I would for sure use experts in the field also.
So then Ulrika, how did you resolve your analysis questions?
Well, it's been a bit of a mix. Mainly we've got help from NMR Center. What happened was that we started off doing a nutritional study in 2015 during cycling. So then I was more interested in some specific metabolites, so I did it myself. But now I get great help from Daniel Malmodin at the NMR Center, who does the bioinformatics analysis.
And Tom, your 90 million data points that you highlighted, how did you make sense out of them?
Well, we have a very skilled bioinformatics team in our group, and of course they helped a lot with all the different aspects of analysis we did. But I mean, I mean, since you mentioned a lot of data, I mean, right now the data that has been collected in our projects are on what are they called? Like secure servers in Uppsala. But of course, the management of the data is getting increasingly more sustainable.
So Eva, you're eager to go next. Go ahead.
No, I just wanted to say that we have done both, a combination, but for the big transcriptomic project, we have got the support from NBIS. So that has been very good. But I would also like to point out that if you're working with a complex disease and a biological concept, it's not enough to have any bioinformatician. You must have a bioinformatician that also gets into the research questions and the biology. And that's why generic bioinformatics is not always the best. You have to establish a long-standing collaboration so that you start talking the same language. I think that is important, at least from my aspect.
very good point. And Kristoffer, do you have something to add here?
No, I've kind of done the same. So we started out quite early with generating large data sets, and I also learned in the beginning, but then it got out of hand quite quickly. So that's why some of the projects just took a long time to actually get going before I could get people that could properly analyze them. So now I actually have 2 bioinformaticians in the group and another one which will learn to do bioinformatics as his project progresses. Because it's so important to have these individuals that also understand, I think, the biology of it. So having a long-standing collaboration is really important.
Yeah. No, I agree with all of you, and that's also my impression, that you need some expertise yourself. Sometimes you DIY because you need to understand your data, but then quite quickly it becomes unmanageable for a single research group. And I guess this is also partly why we have the new program of data-driven life science, because this is so much needed for any future research. But with that, once more, thank you for coming to our outreach day, for presenting, and for all of you in the audience joining. I know there will be an opportunity to have breakout rooms if you have specific questions for the technology experts, so the platform representatives, regarding specific methods that will be available if you're interested once we close this session in 3 minutes. So do stick around. I'm pretty sure there are representatives from all the 7 platforms highlighted today. And of course, the speakers, feel free to hang around as well if you have the time, if people have more questions. And of course, look at the SciLifeLab homepages. Don't be a stranger. We are easily reachable people by email or phone. And really, thank you all for coming today for this first session. Okay. Hej då.
Hej då. Thank you.